Okay, ready. Take 105. Let's go. By the way, just wanted to point out Mr.Bird soap. Hey, this smells really good. Put that back. Okay, so today we're gonna do cellular respiration review. So let's talk about glucose breakdown. The first stage in glucose breakdown is glycolysis, of course. Glyco, which is Greek for sweet, and lysis, which is for split. It's like a split. Glycolysis is the splitting of glucose, obviously, if you put that together. Glucose is a 6-carbon sugar. It is split into 2 pyruvates. Pyruvate is a 3-carbon sugar. Glycolysis happens in the cytoplasm. Glycolysis makes 4 ATP, but 2 of them are used in the process, so only 2 ATPs are saved, sadly. So after glycolysis, there are 2 options for the pyruvate. If there is oxygen present in the pyruvate, it goes through aerobic, which is with air respiration. Aerobic respiration takes place in the mitochondria, also known as cellular respiration. If there is no oxygen present in the pyruvate, cannot enter the mitochondria. Instead, it goes through anaerobic respiration, which means without oxygen respiration. It also takes place in the cytoplasm. There are 2 types of anaerobic respiration, also known as fermentation. There's alcohol fermentation, which usually plants use, like yeast, and there's lactic acid fermentation, which we use as animals. Lactic acid fermentation is another reason why you get sore while you're running or like after a workout. It's because of lactic acid fermentation. So let's talk about the 2 parts of glycolysis. There's glucose activation and energy harvesting. For glucose activation, there's 2 phosphates are added to the glucose molecule. The 2 phosphates come from 2 ATPs. The phosphorylated glucose molecule is called fructose bisphosphate. Because fructose bisphosphate is unstable, it breaks apart into 2 G3Ps. G3P stands for glycerolide 3-phosphate. I don't even know how to say that. The 3 is because of the number of carbon it is. So next, let's talk about energy harvesting. The 2 G3Ps lose their high energy phosphate, releasing 2 ATPs each. Because there are 4 ATPs made and 2 ATPs used during the activation process, we say there's a net gain of 2 ATPs. The two G3Ps that lost their phosphate are now called pyruvates. The goal of glycolysis is to make the 2 pyruvates so they can enter the mitochondria, which is vital to you breathing. So in order for you to enter the mitochondria, of course, and begin cellular respiration, there must be oxygen present. If there is oxygen present in the process known as aerobic respiration, as I said earlier, if there is no oxygen present in the pyruvate, it cannot enter the mitochondria, so it's called anaerobic respiration. So now let's talk about the Krebs cycle. The Krebs cycle, first of all, can also be called the citric acid, because citrate is the first molecule produced in the cycle. The first stage of aerobic respiration is the pyruvate is split to form CO2 and acetyl group, and an acetyl group. The acetyl group attaches to CoA to make acetyl CoA. Acetyl CoA enters the mitochondria along with NADH that was produced from glycolysis. The second stage of the Krebs cycle, so the acetyl CoA that donates its acetyl group to the 4-carbon molecule. So then the CoA is now released, and then H2O then is donated in hydrogen to the CoA molecule and oxygen to the citrate. Citrate is rearranged to form isocitrate. Isocitrate forms alpha ketoglutarate by releasing CO2. Two electrons, so 2 and another 2 electrons, and then H plus are captured by HAD plus to make NADH. So after that, the alpha ketoglutarate forms succinate by releasing CO2. 2 new energetic electrons in H plus are captured by another NAD plus to make another NADH. At this point, all 3 carbons from the pyruvate have been released to CO2. Okay, so now succinate is converted to malate, which contains 2 additional hydrogens and 1 additional oxygen from H2O. Malate is converted to oxaloacetate. I don't know how to say that. I apologize. So, and then 2 more energetic electrons at H plus are captured by NAD plus to make another NADH. So that's the 3rd one. So, and at the end of this all, there are a total of 10 NADH and 2 FADH2 have been created. This is the main source of ATP. This is how they get their ATP. The NADH and the FADH are going to transport H plus through the mitochondrial matrix so they can create H plus concentration gradient. Through the process of chemiosmosis, the H plus are going to push through the ATP synthesis, generating the majority of the ATP. And that's how we know the Krebs cycle. Thank you. Have a nice day. Be sure to like, share, subscribe, comment down below, do everything. You know, also want to compliment Mr. Bird again on the soap. It smells really good. I'm going to put that back. Okay. Peace.